Tejas, can you hear me?
Yes, sir.
Okay. So last class हम डिस्कस कर रहे थे gene and its regulation. The gene and its regulation of the gene expression. स्क्रीन शो हो रही है तुम्हें? In last class, we discussed about the lack operon, and the lack operon was also a result of the close association between the geneticists like Francis Jacob and the biochemist Jacob Monod. तो ये बात याद रखना, lack operon was observed by the firstly observed by the proposed by the Francis Jacob and the biochemist Jacob Monod. And they were first to elucidate a transcriptionally regulated system. हमने लास्ट क्लास में पढ़ा था कौन से लेवल है यहाँ पर, ट्रांसक्रिप्शन लेवल, मतलब यू के अगर हम बात करें प्रोकैरियोटिक सेल में, इफ वी टॉक अबाउट द प्रोकैरियोटिक सेल, so we can regulate the gene expression at the level of the transcription. That means the formation of the primary transcript such as the mRNA from the DNA. So in this lag operon we regulate the gene expression at the time of the transcription transcription level. That's why known as the transcriptionally regulated system. In lag operon, their lag refers to the लास्ट क्लास हमने डिस्कस भी किया था, लैक मींस द लैक्टोज, a polycystic structural gene is regulated by the common promoter and the regulatory genes. Common promoter and the regulatory gene. And such arrangement is very common in the bacteria and referred to as operon. Very common into the bacteria. Is the operon. To name few such example like operon, जैसे कि कुछ operons हैं, like lac operon है, ट्रिप्टोफैन ओपेरॉन है, अर्जिनिन ओपेरॉन है, हिस्टिडीन ओपेरॉन है, and then valine operon. The lac operon consists of the one regulatory gene such as the I gene. Here the term I does not refer to the inducer. इसका I का मतलब यहाँ पर ये नहीं है कि ये does not represent the inducer but derived from the word inhibitor. तो ये प्रेजेंट कर रहा है वर्ड किसको, आई जीन इनहिबिटर को. एंड थ्री स्ट्रक्चरल जीन जेड वाई ए, एंड तीनों स्ट्रक्चरल जीन का क्या है, अलग अपना एक रोल होता है, अलग सिक्रेशन होता है. Such as the I gene, जिसको हम क्या बोल रहे हैं, इनहिबिटर जीन. I gene codes for the repressor of the lac proton. Very very important point. I genes code for the repressor of the lac operon, and the Z gene codes the beta galactosidase enzyme, which is responsible for the conversion of the lactose into the galactose and the glucose, which is primarily responsible for the hydrolysis of disaccharide lactose into its monomeric units such as the galactose and the glucose. अभी बताया मैंने. And the Y gene codes for the permease enzyme, which increase the permeability of the cell to बीटा गैलेक्टोसाइडेज. एंड Y gene क्या करेगा, जो परमेबिलिटी है सेल की उसको क्या करता है, इंक्रीज करता है, जिसकी वजह से क्या होगा, ड्यू टू दिस increase permeability of the cell to beta galactosidase, and beta galactosidase enters into the cell. And the A gene ZYA, Z हो गया, Y हो गया, and then A gene encodes a trans-acetylase. Hence all the three genes product in the lac operon are required for the metabolism of lactose. In most of other operons, as all genes present in the operon are needed together to function in the same related metabolic pathway. Important point नहीं है. Like lac operon क्या है, promoter, inhibitor, I gene जो है ये, and the again promoter, and then this one is the operator ZYA. ये क्या है, तीनों स्ट्रक्चरल जीन है. इसका सीक्वेंस याद रखना, PIPO ZYA in the absence of inducer. So क्या होगा, I gene code the repressor mRNA. repressor mRNA, repressor आया यहाँ पर, and then the bind with the operators, and repressor Repressor binds to the operator region and prevent the RNA polymerase from transcribing the operon. When the I gene codes the repressor mRNA, repressor which bind to the operator region, this one, इससे क्या होगा, ये bind होगा and prevent RNA polymerase from the transcribing the operon. और RNA polymerase क्या हो, bind नहीं होगा किससे, promoter से. अब इन द प्रेजेंस ऑफ इंड्यूसर, यहाँ पे इंड्यूसर क्या है, यहाँ पे इंड्यूसर है क्या, लैक्टोज. व्हेन इन द प्रेजेंस ऑफ द इंड्यूसर, मतलब लैक्टोज की प्रेजेंस में अगर हम बात करें, सो रिप्रेसर आरएनए क्या हुआ, कोड हुआ. इस रिप्रेसर आरएनए से कौन बाइंड करेगा, इंड्यूसर. एंड इंड्यूसर क्या है यहाँ पर, लैक्टोज. एंड व्हेन द इंड्यूसर बाइंड टू द रिप्रेसर एमआरएनए, दैट्स is convert into the active repressor to the inactive repressor and can't bind with the operator region, तो इससे बाइंड नहीं हो पाएगा. एंड देन जेड बाई ए लैग एमआरएनए ट्रांसक्राइब हुआ. बीटा गैलेक्टोसाइडेज जेड वाई से, जेड से वाई से क्या, परमिएज, इन्हें से ट्रांसएटाइलेज which is responsible for the translation. And the beta galactosidase responsible for the digestion of the lactose into the galactose and as well as lactose, मतलब I mean glucose. And then permease, permeability increase करता है. Transacetylase is genes for the transacetylase. So look here, lactose is a substrate for the enzyme beta galactosidase and it regulates switching on and the off of the operon. And system as the inducer. In the absence of preferred carbon source, such as the high glucose, if lactose is provided in the growth medium, if lactose is provided in the growth medium of the bacteria, that lactose transport into the cell through the action of permeability, permease. तो permeability को क्या करा, इसकी increase कर दिया. And lactose then induce the operon the following manner, and the repressor of the operon is synthesized. Repressor of the operon is synthesized all the time, continuously formed from the gene. The repressor protein binds to the operator region of the operon and prevent. The repressor protein bind with the operator region to the operon prevent. In RNA polymerase from the transcript the operon in the presence of the inducer, such as the lactose or allolactose. The repressor is inactivated by the inactivation with the inducers. And this allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of lag operon can also be visualized as regulation of enzyme synthesis by its substrate. Then, glucose and galactose cannot act as the inducer for the lag operon. The answer of the lag operon, like regulation of lag operon by repressive is referred as the negative regulation. If the regulation of lag operon with the help of the repressor, referred to as the negative regulation. Lag operon is under control of the positive regulation as well, but it is beyond the scope of discussion at this level. Regulation of the leg upon by repressive is referred to the negative regulation. So this one is the leg operon. ये हो गया ह्यूमन जेनेटिक्स मोनो एंड मल्टीफैक्टोरियल कैरेक्टर ट्रांसलेशन मेनली ये हम डिस्कस कर चुके हैं इन द इनहेरिटेंस ऑफ वन जीन एंड द सेकंड जीन भी तो मेनली आज हम डिस्कस फोकस करेंगे हेरिडिटी एंड डिजीज पर ऑटोसोमल एंड द लिंक टू द क्रोमोसोम एक्स कौन कौन सी डिजीज होती है किसी डिजीज जो है हमारी जो है यूनिट कंप्लीट होने वाली है Human Genome Project (HGP) was called a mega project. You can imagine magnitude requirement for the project, such as the Human Genome Project said to have approximately 3 into 10 power 9 base pair. And the cost of sequencing required $3 per base pair. Estimated cost in the beginning. Total estimated cost of the project would be approximately $9 billion. Human Genome Project, $9 billion. And if the obtained sequences were to be stored in typeform in books and each page of the book contained 1000 letters and each book contained 1000 pages. Each page भी कौन सी है? Like contain 1000 letters हैं एक पेज पर. कंटेंट कितना होंगे 1000 पेज ही होंगे and then 3300 such books would be required to store the information of DNA. Required to store the information of DNA sequence. So here is the sex-linked inheritance. One of the earliest known as the Institute of Sex-linked Character is the bleeder's disease like hemophilia. And hemophilia is the type of the sex-linked inheritance. Always remember that observed only in the males in the royal family of the Britain. Royal family of the Britain. The concept of sex-linked inheritance was introduced by Thomas H. मॉर्गन। सबसे पहले जो कॉन्सेप्ट है हीमोफीलिया को किसने दिया? थॉमस एंड मॉर्गन। इन द 1910 while working on the Drosophila melanogaster लिंकेज और निकोमलेस जब हमने पढ़ा था। Firstly is the sex determination. And as we already know about the law of segregation, at the time of the gametogenesis, both pair of the chromosomes separate or segregate from the each other, such as the allele also segregate or separate at the time of the gametogenesis, and gametogenesis take place at the time of the meiosis cell division. That's why in the meiosis cell division, from number of chromosomes in the daughter cell will be half to their parental cell. So diploid cell transform into the four haploid daughter cells. The sex chromosome prepare XY and X type of the XY. But in both cases, male produce different types of the gamete. Either with or without X chromosome, and some gamete with X chromosome, and some with the Y chromosome. And such type of sex detection mechanism, the male heterogamety and the female heterogamety. The male heterogamety is like XY. Female heterogamety the sex determination in the human being. If the the sex chromosome on the humans, like we talk about the male, तो मेरे के पास जो सेक्स क्रोमोसोम होंगे 22 पेयर्स प्लस एक्स वाई पेयर ऑफ क्रोमोसोम। हाउ मेनी पेयर? 23 हो गए। फीमेल के पास कौन से पेयर होंगे? 21 प्लस 22 प्लस एक्स एक्स। Is the sex determination in the humans. Now we will talk about the Mendelian disorders. If we talk about the Mendelian disorder such as the Mendel disorder and the chromosomal disorder. And Mendel disorder mainly determined by the alteration of the mutation in the single gene. How we can determine the Mendel disorder? Because of the due to the alteration or change or the mutation in the single genes. The disorders are transmitted to the offspring to the same line as we have studied in the principle of inheritance. अब इन ये जो डिसऑर्डर हैं किस वे में ट्रांसमिट होंगे? टू आर ट्रांसमिटेड टू द ऑफस्प्रिंग ऑन द सेम लाइंस एज वी हैव स्टडीड इन द प्रिंसिपल ऑफ इनहेरिटेंस. And the pattern of the inheritance of such Mendelian disorder can be traced in the family by the pedigree analysis. Even the pattern of the inheritance of such Mendelian disorder can be traced in the family by the pedigree analysis. The most common type of the Mendelian disorder is the हीमोफीलिया, cystic fibers, fibrosis, sickle anemia, cell anemia, and the color blindness and phenylketonuria and thalassemia. So, dominant recessive similarly, trait may also be linked to the sex chromosome in the case of the hemophilia. The trait है, which can also bind a link to the sex chromosome. In a case of hemophilia, it is evident that this X-linked recessive. So first one is the hemophilia. And this sex-linked recessive disease. Which type of the disease? Sex-linked disease. Very, very important point. We saw the transmission from unaffected carrier. So the transmission from unaffected carrier female to some of the male progeny. That means in which so its transmission from unaffected carrier female, unaffected carrier female to some of the major progeny has work or has been widely studied. The next point is the cascade of the protein ball in the clotting of the blood is affected, and due to this, in affected individual is a simple cut will start in non-stop. First compare family degree chart. Sky Queen Victoria. So number of hemophilic and then sickle cell anemia. And this is autosome linked sex. Hemophilia is the sex linked recessive disease. But if we talk about the sickle cell anemia, this is an autosomal linked recessive. And trait can be transmitted from parent to offspring. Sickle cell anemia. This is known as the autosomal linked recessive trait that can carry for a gene or the heterozygous. Can carrier or the heterogeneous carrier. And the disease is controlled by a gene pair of allele, HBA, HBB, out of three possible genotypes. Only HB and the HS. Out of the three possible genotypes, for only homozygous individual for HBS and the HBS2 and so the disease phenotype. So the respect to the disease helpful, no, not helpful. So the disease phenotype undergoes SBA and the HBS. Individual appear between unaffected, but there 50% of probability of transmission. The total gene to the progeny, thus exhibiting such an animal cell, must detected further by the substitution of human genome. Like at the time of the analysis. So this one is the normal cell and this one is the sickle cell animal cell. नॉर्मल सेल की जो शेप होती है वो क्या होती है लेस देन द एक मिनट। हाँ तेजस कुछ बोल रहे हो? अगेन यहाँ पे देखो, एक बार हम बात करें सिकल सेल एनीमिया की। सिकल सेल एनीमिया इज ऑटोसोमल लिंक डिजीज that can be transmitted from parent to the offspring when both parents are carrier for a gene or the heterozygous. And this disease is controlled by the disease single pair of the allele such as the HB and HPS. And out of three possible genotypes, only homozygous individual, which is the HPS, like HPS, ये हो गया homozygous condition में डिजीज फिनोटाइप। इन जो हेटेरोजाइगस कंडीशन है एच बी ए एंड एच पी एस, देखो यहाँ पर क्या है, अलीलिक पेयर जो है वो अलग अलग हैं, एक है एच बी ए एंड एक है एच बी एस। ये कौन सा है? हेटेरोजाइगस। एंड दिस वन इज द एच बी एस एंड द एच बी एस, दिस इज द होमोजाइगस इंडिविजुअल। इंडिविजुअल अपीयर apparently unaffected but they are carrier for the disease as the 50%। अब जो हेटेरोजाइगस वाले जो होगा, इंडिविजुअल अपीयर apparently unaffected, बट ये जो हेटेरोजाइगस कंडीशन है, क्या अनअफेक्टेड तो होगा बट कैरियर होगा ये, which can transmit the disease to another person, another offspring. A carrier of a disease has a 50% of the probability of the transmission of human gene to progeny. Thus exhibit the sickle cell trait. The defect is caused by the, ये बहुत इंपॉर्टेंट पॉइंट है यहाँ पर सिकल सेल एनीमिया में, डिफेक्ट। द डिफेक्ट इज caused by the substitution of the glutamic acid by the valine। किस पोजीशन पर हो रहा है ये? 6th पोजीशन पर। सच एज द लुक हियर, नॉर्मल हीमोग्लोबिन है, शेप की होती है इसकी बाइकॉनकेव होती है, एंड इसमें जो कॉम्प्लीमेंट्री बेस सीक्वेंस वो क्या है? GAG। CTC, CTC का क्या होगा यहाँ पर? GAG आ गया। GAG कोड्स द जेनेटिक कोडोन, GAG जो होगा उसमें, GAG कोडोन कोड द ग्लूटामीन, ये ग्लूटामिक एसिड क्या करता है? कोड करेगा। बट एट द ड्यू टू द सम फैक्टर्स, सिकल सेल एनीमिया में जो GTG एंड द CAC है हमारे नहीं, तो सीक्वेंस क्या होता है? चेंज होता है। एक ही जगह यहाँ पर क्या होगा? U, बिकॉज ऑफ थायमिन एब्सेंट इन द एमआरएनए, बट एडेनिन तो प्रेजेंट होता है एमआरएनए में, तो थायमिन ओनली बाइंड विद द एडेनिन। पर यहाँ पर क्या है? ये डीएनए हो गया एंड दिस वन इज द एमआरएनए। एमआरएनए यहाँ पर क्या है? GUG, बिकॉज ऑफ द कॉम्प्लीमेंट्री प्रिंसिपल एट द टाइम ऑफ द आरएनए, because as you already note at the adenine only combined with the uracil and the cytosine combined with the guanine, but in the case of the eukaryotic cell, adenine only combined with the thymine and thymine combined with the adenine। पैरामिडिन में दोनों में क्या होगा? सेम होगा। सो 6th पोजीशन में जो ग्लूटामिक एसिड है वो क्या क्रिया हो रहा है यहाँ पर नॉर्मल नॉर्मल आरबीसी में, बाय एट द टाइम ऑफ द सिकल सेल एनीमिया 6th valine, 6th valine instead of the glutamic acid, and due to this valine, the 6th position of the beta globin chain of the hemoglobin molecule, the substitution of amino acid in the globin proteins results to the single base substitution at the 6th codon of the beta globin gene from the GAG and to GUG। Then mutant hemoglobin molecules, hemoglobin molecule से mutant जो हुआ है, undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from the biconcave disc। तो उसकी जो होगी लो ऑक्सीजन टेंशन होगी ड्यू टू द शेप ऑफ द RBC from the biconcave disc to the elongated sickle-like structure, such as here, elongated disc-like structure। Last one of the mental disorder is phenylketonuria, and inborn error of the metabolism is known as the inherited as the autosomal recessive disease। First of all, phenylketonuria is the autosomal recessive trait, and the affected individual lacks an enzyme, the affected individual है lacks an enzyme that converts the amino acid phenylalanine into the tyrosine। ये पॉइंट जो है ये है इंपॉर्टेंट से मेन स्टेप की अगर हम बात करें द फेनिक्यूटिनेरिया। तो फर्स्टली जो स्टेटमेंट है जो इंपॉर्टेंट है, अफेक्टेड इंडिविजुअल लैक्स एन एंजाइम दैट कन्वर्ट द एमिनो एसिड फेनिलानिन इनटू द टायरोसिन। एज अ रिजल्ट ऑफ द फेनिलानिन इज एक्यूमुलेटेड एंड कन्वर्टेड इनटू द फेनिल पायरुविक एसिड। Thus, diabetes acts as the accumulation of these in brain result in mental retardation. So right now we will talk about the chromosomal disorders. And the chromosomal disorders, to the other hand, go to the absence or excess or abnormal arrangement of one or more chromosomes. So failure of segregation of chromatids during cell division. Failure of segregation chromatids during the during cell division cycle results in the gain or the loss of the chromosome. क्या होगा? Aneuploidy. Like for example, Down syndrome result in the gain or extra copy of chromosome, which 21st chromosome. Similarly, Turner syndrome and last due to the loss of X chromosome in the human female. And failure of cytokinesis after telophase. So failure of cytokinesis after telophase stage of the cell division results in the increase in a whole set of chromosome in an organism. The phenomenon is called the polyploidy. क्या हो गया? Polyploidy. And the total number of chromosome in जो normal basic हैं जो हम पढ़ चुके हैं already. Total number of the chromosome in the human beings the 46. Out of these 22 pairs are the autosome and the one pair of the chromosome are the sex chromosome, like the X in the female and the XY in the male. Sometimes, so rarely, either an individual copy of the chromosome may be included in an individual, or an individual may lack one of any one pair of the chromosome. इसमें क्या है? कुछ सिचुएशन दी गई है हमें, लाइक ट्राइसोमी और मोनोसोमी of a chromosome respective to the such condition leads to care. Down syndrome, Turner syndrome, Klinefelter syndrome, and the example is the chromosomal disorder. These all are the examples of the chromosomal disorders, such as the Down syndrome, Turner syndrome, Klinefelter syndrome. अब हम बात करते हैं इसकी Down syndrome. So the cause of the genetic disorder in the presence of additional copy of the chromosome number 21. So at the time of the Down syndrome, what will happen? Trisomy of the chromosome take place. What do you mean of the trisomy? Additional copy of the number of chromosome of the chromosome number 21 की additional copy. First described by the Langdon Town, 1866. The affected individual is short, statured with small round headed furrowed tongue and the partially open mouth. मेनली जो इम्पोर्टेंट है यहाँ पर वो है क्या? Klinefelter syndrome. Genetic disorder is also called due to the presence of the additional copy of S chromosome, resulting into the karyotype of the 57 X chromosome, resulting into the karyotype होगा इसका 47XXY. ये chromosome है. Such an individual has overall masculine development, however the feminine development of breast. Gynecomastia is also expressed figures, and such individuals are sterile. Last one is the Turner syndrome. So such a disorder is a cause due to the absence of close together Turner syndrome. Such a disorder is the cause due to the absence of one of the X chromosome, such as the 45 with exotype female. Our sterile ovaries are rudimentary beside other features, including this of the other sexual character. ये यूनिट जो है वो कंप्लीट हो गई है. आगे हम आते हैं बायोटेक्नोलॉजी, रिकॉम्बिनेंट डीएनए टेक्नोलॉजी एंड इट्स एप्लीकेशन. Wait 2 minutes. Hereditary disease, autosomal, and the link to the chromosome, which link to the chromosome X. कोई डाउट इसमें किसी को, जो हमने अभी डिस्कस किया आज था?
नो सर।
तेजस,
Yes, sir.
any doubt?
No, sir.
Okay. So now we will talk about the biotechnology and the recombinant DNA technology, its application. So can you see screen? Tell me
Yes,
fast.
sir.
Okay. So now we will talk about the biotechnology, the principle and their processes. So firstly, what's the mean of the biotechnology? तो उसकी एक बहुत अच्छी डेफिनेशन है इन द आवर एनसीईआरटी बुक. Look here. Biotechnology. Biotechnology deals with the technique of using live organism or enzyme. Yeah. So biotechnology deals with the techniques of using live organism or enzyme from organism to produce product and the process useful to humans. Like क्या है कि एक branch of science है in which the using living organism or their enzyme the secret from the organism to produce product and processes which useful to humans is known as the biotechnology. And according to the अगर हम बात करें examples of the biotechnology such as the in vitro fertilization, IVF. So fertilization takes place outside the body, leading to a test tube baby, synthesizing a gene using it, developing a DNA vaccine, correlating or correcting of a defective gene. All are the part of biotechnology. And according to ये जो definition है, according to European Federation of Biotechnology (EFP), given a definition of the biotechnology that according to the IMAT exam, this definition is the important. IMAT according. Says the European Federation of the Biotechnology (EFB) has given a definition of the biotechnology that encompasses both traditional view and the modern molecular biotechnology. Like अगर हम बात करें traditional view की, traditional view of the biotechnology, what will be? Such as the preparation of the curd, the these all are the traditional view of the biotechnology. And if we talk about the molecular biotechnology, the definition given by the if we देखो molecular अगर हम बात करें दोनों के view से, the integration of the natural science and organism cells part thereof and the molecular analogs for products and their services known as the so here integration of natural science and organism cells and parts thereof and molecular analog for product and services. So firstly, principle of the biotechnology, genetic engineering. तो मेनली जो हमारे जो मेन इसमें गोल रहता है या फिर मेन एम जो होगा, मेन अप्रोच जो रहेगी बायोटेक्नोलॉजी में, वो क्या है, हम करते क्या इसमें बायोटेक्नोलॉजी. तो टेक्निक्स टू अल्टर द केमिस्ट्री ऑफ द जेनेटिक मटेरियल सच एज द डीएनए एंड द आरएनए. फर्स्ट पॉइंट प्रिंसिपल है क्या हमारे बायोटेक्नोलॉजी का, तो इसमें मेन अप्रोच जो रहेगी इसमें क्या करते क्या हम, टेक्निक टू अल्टर द केमिस्ट्री ऑफ द जेनेटिक मटेरियल डीएनए एंड द आरएनए and introduce into the host organism, thus change the phenotype of the host organism. Second is the bioprocess engineering, maintenance of the sterile microbial contamination free ambience in the chemical engineering process to enable the growth of the only desired products. Desired microorganism egotic cell in the large quantity of the manufactured products. आगे हम बात करें, look here, recombinant DNA. This is the very important. Useful of the gene cloning and the gene transfer. So mainly, what's the mean of the recombinant DNA? So firstly, tell me about the, do you know about the plasmid?
Yes, sir.
Yes, sir.
So what is the plasmid?
Plasmid is a
Circular
self-replicating
DNA.
circular DNA.
Extra chromosomal DNA which is present in the bacteria. Is it clear? which can self replicate to itself. Plasmid. So when the human gene या another DNA combine with the plasmid, क्या इसमें प्लाज्मिड प्लस another gene, अब जो डीएनए फॉर्म हुआ उसको हम क्या बोलेंगे? रिकॉम्बिनेंट डीएनए. लाइक during the preparation of the insulin hormone, so we extract the insulin gene in the from the human DNA and then cuts the DNA with the help of the enzyme and then bind to the plasmid after the cutting of the plasmid by the enzyme. And the plasmid और जो ह्यूमन जीन है इंसुलिन जीन को हम क्या करते हैं? कंबाइन कर देते हैं विद द हेल्प ऑफ द डीएनए लाइगेज एंजाइम. अब जो फॉर्म हुआ स्ट्रक्चर, कॉम्प्लेक्स जो स्ट्रक्चर में फॉर्म हुआ उसको हम क्या बोल रहे हैं? रिकॉम्बिनेंट डीएनए. उस रिकॉम्बिनेंट डीएनए में जो जीन क्लोनिंग है, रिकॉम्बिनेंट डीएनए क्या करेगा? प्लाज्मिड अपने आप को रेप्लिकेट करता है. अभी हमने पढ़ा सेल्फ रेप्लिकेटिंग है. एंड देन ड्यू टू द सेल्फ रेप्लिकेटिंग the gene will also multiply, and cloning is the copy and the gene transfer. To overcome this limitation has allow us to isolate and introduce only one or a set of the desirable genes without introducing undesirable genes into the target organism. The origin of the replication पढ़ चुके हैं हम ऑलरेडी. Where the replication start? The origin of replication कहां पर होगा? On the plasmid. यहाँ पर थ्योरी लिखी हुई है. पढ़ लो. Do you know about the likely fate of the piece of DNA which is somehow transferred into the aligned organism? Like most likely this piece of the DNA would not be able to multiply itself. हम ऐसा recombinant DNA की हमें जरूरत क्यों? Why we need to the formation of the recombinant DNA? Because of the piece of the DNA can't replicate itself in the progeny cell of the organism. But when it get integrated into the genome of the recipient, the genome है recipient का, like plasmid, then it can multiply and inherited along with the host DNA. This is because the aligned piece of DNA has become part of a chromosome which has ability to replicate. Ability to replicate. In a chromosome there is a specific DNA sequence called the chromosome में क्या होता है? एक specific DNA sequence होता है जिसको बोलते हैं origin of replication, which is responsible for the initiating replication. पढ़ चुके हैं already. And therefore for the multiplication of any aligned piece of the DNA in an organism, it needs to be part of the chromosome. An aligned DNA linked with the origin of replication, and thus aligned piece of the DNA can replicate and multiply itself in the host organism. इसको क्या बोल रहे हैं हम? क्लोनिंग. लाइक अब बात करते हैं प्लाज्मिड. तो इन दिस चैप्टर ऑफ द बायोटेक्नोलॉजी टोटली हम डिस्कस करेंगे क्या? रिकॉम्बिनेंट डीएनए. like autonomously replicating circular extrachromosomal DNA which is present in the bacteria like as the Salmonella typhimurium. Stanley Cohen and the Herbert Boyer and accomplished this is the 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from the plasmid, and which is the responsible for the conferring antibiotic resistance. तो 1972 में क्या किया? स्टैनली कोहेन एंड द हार्वर्ड बॉयर ने isolate the antibiotic resistant gene and the cutting out a piece of the DNA from the plasmid. And the cutting of DNA at the specific location become possible with the discovery of the so-called molecular seizures, which also known as the restriction enzymes. And restriction enzyme are of mainly two types, exonuclease and endonuclease. आगे हम बात करेंगे उनकी. So molecular seizure will be restriction enzymes. And the cut piece of DNA was linked with the plasmid DNA. Cut piece of DNA was then linked with the plasmid DNA. जो DNA हमने कट किया, like human insulin gene को हमने कट किया, and the plasmid को हमने कट किया. But always remember that both DNA, human insulin gene वाला जो है DNA, human DNA है, and the plasmid, which is also a DNA, both cut the same type of the restriction enzyme. Because of the, we need to obtain the same sticky ends. Sticky ends क्या होते हैं? Like grips जो होती हैं. And these plasmid DNA act as the vector. Plasmid DNA act as the vector and transfer the piece of DNA attached to it. And you probably know that the mosquito act as the insect vector to transfer the malaria parasite into the human body. ये सब पता है वो तुम्हें. तो मेन चीज है यहाँ पर क्या ये. You can hence infer that there are three basic steps in genetically modify an organism. GMO. Three basic steps होते हैं इसके. First one is the identification of DNA with the desirable gene. Look here. Firstly, we identify the DNA with desirable gene. मतलब हम क्या करेंगे, सबसे पहले आइडेंटिफिकेशन करेंगे डीएनए का. वो भी वो डीएनए जिसमें वो जीन होनी चाहिए जो हमें चाहिए, जिसके हमें डिजायर है. तो उस डिजायर वाले जीन को हम क्या करेंगे, सबसे पहले डीएनए को आइडेंटिफाई करेंगे. लाइक हमें चाहिए इंसुलिन जीन. So we identify, firstly we identified the DNA which have the insulin gene. And after that, cutting the DNA, this desirable gene, and bind with the plasmid, form the recombinant DNA, and introduce into the identified DNA into the host. जो identified DNA है उसको क्या करेंगे, host में introduce करेंगे. और यहाँ पर host क्या होगा, बैक्टीरिया, such as the E. coli, Escherichia coli. And then third step, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Third case में maintenance करेंगे introduced DNA in the host and transfer of the DNA to its progeny. A very important point or topic of this chapter, tools of recombinant DNA technology. And tools में क्या होगा यहाँ पर, some types of the enzymes which is very useful in the biotechnology, such as the restriction enzyme, which is the molecular seizure and tendency to the cutting of the DNA mainly. Polymerase enzymes like DNA polymerase, RNA polymerase. If we talk about the DNA polymerase, mainly responsible for the replication of the DNA. Restriction enzyme, polymerase enzyme. And polymerase enzyme mainly responsible for the replication and the transcription of the DNA. And the ligase enzyme responsible for the joining of the DNA. Vectors like plasmids and the host organism. And host organism कौन है यहाँ पर, bacteria. So firstly, we will discuss about the restriction enzyme. So in the 1963, two enzymes responsible for the restricting the growth of the bacteriophage in Escherichia coli were isolated. तो 1963 में 2 एंजाइम जैसे हमें आइसोलेट तो डिस्कवर किए गए, जो रेस्पॉन्सिबल फॉर द रेस्ट्रिक्टिंग द ग्रोथ ऑफ द बैक्टीरियोफेज, जो बैक्टीरियोफेज की ग्रोथ के लिए क्या थे, रेस्ट्रिक्ट इन द ई कोलाई वर आइसोलेटेड. इसमें आइसोलेट किए गए ई कोलाई से. वन ऑफ दिस एडेड द मैथिल ग्रुप्स ऑफ द डीएनए. ये क्या कर रहा था, मैथिल ग्रुप्स ऐड कर रहा था डीएनए में, while the other cuts the DNA. And which cuts the DNA, बाद में क्या बोला गया, restriction endonuclease. And the first restriction endonuclease, hint second, ये जो पॉइंट्स हैं इस पैराग्राफ में ये बहुत इंपॉर्टेंट है। So first restriction endonuclease, hint second, whose function depends on the specific DNA nucleotide sequence पर was isolated and characterized by the 5 letters. So mainly हमें इतना पॉइंट डीपली नहीं जाना, बस ये पॉइंट याद रखना है हमें। फर्स्ट रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम कौन सा है, हिंट सेकंड, इसका फंक्शन डिपेंड करता है अपने स्पेसिफिक डीएनए न्यूक्लियोटाइड पर, सीक्वेंस पर, was isolated and characterized 5 years later. And the hint second always cuts DNA molecules at a particular point by the recognizing a specific sequence of the 6 base pair. Always remember that. जो हिंट सेकंड रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम है वो फर्स्टली क्या करेगा, रिकॉग्नाइज द स्पेसिफिक लोकेशन, स्पेसिफिक सीक्वेंस ऑफ द न्यूक्लियोटाइड बेस पेयर्स। हाउ मेनी बेस पेयर का वो क्या होगा, स्पेसिफिक लोकेशन होगी 6 बेस पेयर। हाउ मेनी बेस पेयर, 6 बेस पेयर। And then this specific base sequence is known as the recognition sequence for hint second. जबकि अगर हम बात करें hint second, today इसके अलावा अगर हम बात करें, today we know more than 900 restriction enzymes that have been isolated from the 230 strains of the bacteria. Actually, till now, you know about the 900 restriction enzymes. And these all are the isolated from the 230 strains of the bacteria mainly. So, classification of the restriction enzyme, such as the naming की अगर हम बात करें enzyme की, so first letter of the name comes from the genus and the second 2 letter comes from the species. So the first letter comes from the genus, second 2 letter comes from the species of the prokaryotic cell. They were isolated, जिससे isolate किए गए, like eco R1, E. coli जिसको बोलते हैं। E first letter represents the genus, it comes from the genus. CO derived from the species. In the R1 जो R है वो क्या बता रहा है, इसका स्ट्रेन बता रहा है। फर्स्ट जो रोमन नंबर से क्या बता रहा है हमें, इसका ऑर्डर बता रहा है, ये पहले मतलब सबसे पहले कौन सा मतलब हुआ, मतलब डिस्कवर किया गया। सो एस की क्या, कोलाई RY13. सो इन ई कोलाराई द लेटर आर इज ड्राइव फ्रॉम द नेम ऑफ द स्ट्रेन, आर जो है स्ट्रेन बताया मैंने अभी। The Roman numbers along with the names indicate the order in which the enzyme were isolated from the strain of bacteria. Roman number क्या represent कर रहा है, names indicate the order in which the enzyme were isolated from the strain of bacteria. Then restriction enzyme belong to a larger class of the enzyme called the nuclease enzyme. जो restriction enzyme है लार्जर क्लास होते हैं एंजाइम की, इसको क्या बोल रहे हैं हम, न्यूक्लियस एक क्लास होती है, in which the two types of the nuclease enzyme like exonuclease and the endonuclease, and the function will be both enzyme are of same, but location of the action वो क्या होगी, दोनों की वो डिफर करेगी। such as the exonuclease remove nucleotides from the ends of the DNA, ends of the DNA, मतलब जो exonuclease जो DNA को कट करेगा, ये क्या करेगा, एंड से करेगा, मतलब ये वाले पोर्शन होंगे, ये एंड्स, इधर से कट कर दिया, इधर से कट करेगा, कौन करेगा, ये एक्सोन्यूक्लियस। दैट्स व्हाई एग्जिट पार्ट ऑफ द स्ट्रैंड ऑफ डीएनए को क्या करेगा, ये कट करेगा, टर्मिनेटिंग एट। दैट्स व्हाई दिस टाइप ऑफ द न्यूक्लियस एंजाइम इज नोन एज द एक्सोन्यूक्लियस। बाहर की तरफ जो पोर्शन है उनको क्या करेगा, ये स्ट्रेच को डीएनए के स्ट्रेच को कट करता है। बट द एंडोन्यूक्लियस मे कट एट द स्पेसिफिक पोजीशन विद इन द डीएनए, कोई एक स्पेसिफिक लोकेशन को पहले क्या करेगा, रिकॉग्नाइज करेगा, आइडेंटिफाई करेगा, एंड देन ब्रेक द डीएनए, cutting the DNA. In each restriction endonuclease function by inspecting the length of the DNA sequence. And once it finds its specific recognition sequence, it will bind to the DNA and cut the each of the two strands of the double helix at a specific point in their sugar phosphate backbone. क्या करेगा, इसको कट करेगा। And each restriction endonuclease recognizes a specific sequence which is known as the palindromic nucleotide sequence in the DNA. Palindromic sequence that means the which sequence have the same type of the flow when we read the forwardly and the backwardly, such as the GAATTC. अब इसको हम ऐसा पढ़ लें, या इसको हम GAATTC, तो जो सीक्वेंस होगा वो क्या होगा, सेम होगा। खैर बता भाई, एक्शन ऑफ रेस्ट्रिक्शन एंजाइम देखते हैं यहाँ पर जरा। द एंजाइम कट बोथ डीएनए स्ट्रैंड एट द सेम साइट। निको R1 cuts the DNA between the base GNA and only when the sequence GAATC is present in the DNA. So as we already know about that from this point, like eco R1 cuts the DNA between the bases GNA, guanine और adenine के बीच में कट कर, like ये arrow देखो यहाँ पर, arrow represent the action of the eco R1। ऐसे क्या कर कट करेगा, सेम एज लाइक हियर, से भी क्या करेगा, इसको ये कट करेगा। देन ये क्या होंगे, यहाँ से सेपरेट जी, बाकी यहाँ सब एम्प्टी होगा। देन सी, और यहाँ पर सब क्या होगा, ए तक होगा, CTTAA। तो ये जी यहाँ से ये क्या होगा, सेपरेट हो जाएगा। एंड दीज आर द स्टिकी एंड्स, ठीक है, इन्हीं को बोलते हैं कि हम स्टिकी एंड्स। अभी थोड़ी देर पहले हमने डिस्कस किया कि बोथ टाइप ऑफ द डीएनए cuts the same type of the restriction enzyme because of the obtaining the same type of the sticky end। तो इसके जो sticky end है, सेम ऐसे sticky end किसके होने चाहिए, दूसरे वाले डीएनए के, तभी तो लाइगेट करेगा। लाइक सेम इसी डीएनए को हमने, फॉरेन डीएनए को हमने क्या करा, इसको कट किया इन द प्रेजेंस ऑफ eco R1, and after that these are the sticky ends, these are the sticky ends, and bind to there and joined by the DNA ligase enzyme। अब इसी डीएनए को हम क्या बोल रहे हैं, रिकॉम्बिनेंट डीएनए। सो डू यू नो व्हाट पैलिंड्रोमिक्स, हमने डिस्कस किया, these are group of the letters that from the same words with when read both forward and the backward। रीडिंग फ्लो क्या होगा, दोनों का सेम होगा अगर हम बैकवर्डली पढ़े या फॉरवर्डली पढ़े। लाइक मलयालम, तो मलयालम क्या है, इसको हम चाहे ऐसे पढ़ें, फॉरवर्डली ऐसे पढ़ें, या इसको हम ऐसे रीड करें, तो रीडिंग फ्लो होगा वो सेम ही होगा। लाइक हमने क्या पढ़ा अभी, यहाँ देखो, restriction enzyme cuts the strand of DNA at a little away from the center of the पैंड्रोमिक साइट्स। मतलब जो पैंड्रोमिक साइट्स का जो सेंटर है उसके थोड़ा सा उससे क्या होगा, लिटिल अवे क्या करेगा, उसको कट करेगा। बट बिटवीन द सेम टू बेसेस ऑन द अपोजिट स्ट्रैंड, दिस लीव्स सिंगल स्ट्रैंडेड पोर्शन एट द एंड, एंड देयर आर द overhanging stretches called the sticky ends on the each strand. This name so because they form the hydrogen bond and with their complementary cut counterparts. The stickiness of the end facilitates to the action of the enzyme DNA ligase. If the stickiness present on the DNA ends, so DNA ligase enzyme work properly to the ligate the DNA ligate. That means the joining of the DNA. And the restriction endonucleases are used in the genetic engineering to form recombinant molecules of DNA. Restriction enzyme used होगा इसमें genetic engineering to form the recombinant molecules of DNA, which are composed of DNA from the different sources of genome. Look here, इस पॉइंट को हम डिस्कस किया थोड़ी देर पहले ही। When DNA cuts by the same restriction enzyme and the resultant DNA fragments have the same kind of the restriction or some kind of the sticky ends and thus can be joined together end to end by the DNA ligase enzyme। इस डायग्राम के थ्रू हम समझते हैं जरा। First thing is this is the foreign DNA, and we can also say that the desirable DNA, and this one is the vector। Vector क्या होगा, plasmid। The same restriction enzyme cutting both foreign DNA and vector DNA at a specific point. Same restriction enzyme. Cutting both foreign DNA and the vector DNA at the specific point. तो हमने यही पॉइंट पढ़ा यहाँ पर. This one is the when cut by the both same when both DNA cut by the same restriction enzyme, then obtain the same type of the sticky end. तो सेम टाइप ऑफ स्टिकी एंड को ऑब्टेन करने के लिए हमने क्या करा, दोनों ही डीएनए को फॉरेन डीएनए एंड द प्लाज्मिड डीएनए व्हिच इज नोन एज द वेक्टर डीएनए कट बाय द सेम टाइप ऑफ द रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम. And after that we obtain the same type of the sticky ends. Then ये कट हो गया, कट लग गया, and then these sequence of DNA, the gene, bind to the plasmid with the help of the ligase enzyme. Like ligase join foreign DNA to the plasmid DNA. And after the binding of the both DNA, this molecule is known as the recombinant DNA molecule. And then transformation, cell divide, and equal क्या होंगी, ये क्या करेगी, जीन होगी इसमें, वो क्लोनिंग होगी इनसाइड द होस्ट. पहले एक था, देखो दो है, ऐसे ऐसे करके डिवाइड होता रहेगा अगेन एंड अगेन. आस्था
Yes, sir.
समझ में आ रहा है, नहीं आ रहा?
आ रहा है सर.
Okay. So you may have realized that the normally points यहाँ पर, and how to separate and isolate the DNA fragment. फर्स्ट टास्क क्या है हमारा बायोटेक्नोलॉजी में, कि हम डीएनए को सेपरेट कैसे करेंगे एंड आइसोलेट कैसे करेंगे. तो फर्स्टली हमें एक बेसिक क्राइटेरिया पता चल गया बायोटेक्नोलॉजी हम करते क्या है, क्या इसके टूल्स हैं. हमने टूल्स पढ़े यहाँ पर. टूल्स में क्या पढ़ा हमने, एंजाइम्स पढ़े. एंजाइम कौन कौन से हैं, रेस्ट्रिक्शन एंजाइम्स हैं, पॉलीमरेज एंजाइम है, लाइगेज है, वेक्टर्स एंड द होस्ट ऑर्गेनिज्म. दीज आर द ऑल द टूल्स ऑफ द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी या फिर बायोटेक्नोलॉजी. look here, separation and isolation of the DNA fragment. And तुमने पढ़ा होगा जेल इलेक्ट्रोफोरेसिस, in which the separation of the DNA fragments according to their density, heavier than the light year. So the cutting of DNA by the restriction nuclease resulting in the fragments of DNA, and these fragments can be separated by the technique gel electrophoresis. So we can separate the DNA fragments with the help of the gel electrophoresis technique. Since DNA fragments are negatively charged molecule, as we already know that the fragments of DNA, every charge of the DNA is negatively charged, and they can be separated by forcing them to move towards the anode under the electric field through the medium matrix. अब जो नेगेटिव चार्ज है तो ऑब्वियसली एनोड की तरफ क्या होगा, मूव करेगा. यस और नो?
Yes, sir.
And then DNA fragments separated dissolve according to their size, like density, through sieve effect providing by the agarose gel. And इस जेल को याद रखना, which is produced or obtained from the red elegans. And the smaller the fragment size, farther it the moves. और आगे फास्ट में मूव करेंगे. लाइक यहाँ पर हम देख लेते हैं, इसमें देखो जरा, ये क्या है, डीएनए फ्रेगमेंट्स हैं, एनोड कैथोड, and these DNA fragments move toward the anode positive side, and then जो लाइटेस्ट डीएनए होंगे जिनकी डेंसिटी कम होगी, जो स्मॉल साइज के होंगे वो क्या होंगे, आगे होंगे, एंड द जो हैवियर डीएनए फ्रेगमेंट्स होंगे वो क्या होंगे, वो पीछे रहेंगे. So according to the density, we can identify or the isolate or the separate out the DNA sequence, the DNA fragments. So separated DNA fragments can be visualized only under, पर हम इनको only visualize कर सकते हैं कब, वो भी कब, when the after the staining the DNA with a compound known as the ethidium bromide. And हमें bands मिलते हैं क्या, ऑरेंज कलर के, इन द यूवी रेडिएशन. तो मीडियम क्या है यहाँ पर, एथिडियम ब्रोमाइड इन द एक्सपोजर टू यूवी अल्ट्रावायलेट रेडिएशन. एंड यू कैन नॉट सी प्योर डीएनए फ्रेगमेंट्स इन द विजिबल लाइट विदाउट स्टेनिंग. So you can see bright orange color band of DNA in a ethidium bromide stain gel exposed to UV लाइट. तो हमें जो इलेक्ट्रो प्रोसेस के टाइम पर डीएनए फ्रेगमेंट्स ऑब्टेन्ड होते हैं, विच कलर, ऑरेंज कलर की बैंड्स होती हैं, बट इन द एथिडियम ब्रोमाइड स्टेन गेट एक्सपोज टू यूवी लाइट. इन द सेपरेटेड बैंड्स ऑफ डीएनए आर कट आउट फ्रॉम द एग्रोज जेल. डीएनए को हम क्या करेंगे, सेपरेट करेंगे फ्रॉम द एग्रोज जेल एंड द एक्सट्रैक्ट फ्रॉम द जेल पीस, एंड दिस स्टेप इज नोन एज द एल्यूशन. The DNA fragment purified in this way are used in the constructing recombinant DNA. Okay. So आज की क्लास में इतना ही. नेक्स्ट क्लास में हम इससे आगे पढ़ेंगे. ठीक है? एनी डाउट इन टुडे सेशन?
No, sir.
क्लियर है सब?
Yes.
Okay. So you can now end the meeting.